Hi, Michelle Glass here, and welcome to our first video in our Chapter 4, Lecture 4 series, and our topic for this whole chapter are cells. We introduced cells back in Chapter 1 as the smallest living unit of life. So remember, the significance of the cell is that it has all of those different properties of life, and we do see that all living things are composed of cells. But they are small. In fact, cells are microscopic. So to begin our conversation, we need to talk just a little bit about how biologists first identified that living things were composed of cells and how we study these cells. So microscopes first became available in the 1600s, and that's when we really first began to see that living things were composed of cells. In about 1665, Robert Hooke is the person responsible for coining the phrase cells. So he actually observed some oak bark. Plant cells are these rigid rectangular structures because they have cell walls. And so it looked like these small rooms underneath the microscope, maybe like a room that a monk would live in, and those rooms are called cells, or think of a jail cell, which we probably can associate better. And so that's how cells got their name. Even though Robert Hooke is responsible for really naming the cell, and is sometimes described as the father of microscopy, we have Antony van Leeuwenhoek, who actually had better equipment, and he looked at more variety of specimens, and he's contemporary with Robert Hooke. So we see that both of these researchers were important in these advancements in using microscopes and looking at things under the looking at living specimens. By the mid-1800s, enough researchers had viewed enough specimens to develop what is called the cell theory. Remember, a scientific theory is based on a large body of evidence, and so far, all of the data is pointing to this theory as being correct. So we have all of the data that's been collected from 1600s to 1800s to today to demonstrate that all living things are made of cells, and that that cells give rise to new cells. So all cells are coming from other cells. So this is really what we describe as our cell theory. Now, of course, because our cells are microscopic, we do need to use microscopes in order to really investigate these, and there are several different types described for us in our manual. So we have what are called light microscopes. These are the types of microscopes that we have in our labs here, so you're getting to use those this semester. They're using light to illuminate the specimens. There are two types. You have used in your labs both the compound light microscope, that's the microscope that can magnify objects from 40 times all the way up to 1,000 times. And then we have what's listed in our text as the stereo microscope. My training is to call it the dissecting microscope, so that's often how I refer to it. And these can range from 10x to 40x. The microscopes that we have in our labs, I think, just go up to 30 times. So it's not quite as magnified. Stereo microscopes or dissecting microscopes are really good for looking at the surface of a small object, while those compound light microscopes can magnify more. So very often you're looking at a thin slice or thin specimen that you're able to get more detail. Researchers also use electron microscopes, and you'll see pictures of these in your textbook. Pictures of specimens magnified using electron microscopes in your textbooks. These are using electrons rather than light, and so we get much more magnification and much more detail. There are two types of electron microscopes. We have what's called the transmission electron microscope. You can abbreviate that as TEM. This style of microscope is used for detecting internal structures, so that'd be kind of analogous to the compound light microscope, but way more magnified. And then we have what's called the scanning electron microscope, or SEM, and this is going to allow you to see those topographical or surface structures of organisms. So again, this would be similar to like the stereo microscope in its use, except way more magnified. So the next question really then is, well, why are cells so tiny? Is there a limit to the size of the cells? And we see that cells are small actually in order to maintain a high surface to volume ratio. So the surface is talking about that outer border of the cell, and the volume is talking about all the space inside of the cell. Now, if you have a really big structure, we'll see that there's a lot of traffic going across the cell membrane, which is our outer border, but it's going to be hard for that material to actually get to the center of the cell, or from material like waste material from the center of the cell to get out. So if you have a really big cell, it's going to take a long time, and it's going to be hard for stuff to go from the outside to the center, or from the center to the outside. So one of the ways to take care of that is to have, this same kind of sized organism, but composed of a lot of small units. When you make it composed of all these small units, then you're making more surface area, although you might still have the same amount of volume. So you're now increasing your surface area to volume ratio. So now, because you have a smaller structure, it's a lot easier to get from the center to the outside versus when we looked at this particular image. So one of the key reasons that cells are small is to increase the surface area to volume ratio. And then we'll see too that sometimes cells are small just because it's part of their job function. So we'll see that as well, but this is really our key reason. Okay, so that is it for now, and stay tuned for our next video.